Hello everyone, I am Nimanthi Abeyrathna from Clarkson University, New York. Today I am going to present one component of my PhD project, which is morphogenetic characterization of invasive snail Viviparous georgianus in the New York Great Lakes Basin. To start with the background, freshwater ecosystems are diverse ecosystems which are vital for the survival of many organisms. Aquatic invasive species or AIS are a threat to global aquatic ecosystems, and aquatic invasive mollusks plays a major role in that. Introduced species rarely become invasive. And to become invasive, they should first survive in the transportation pathway and next survive in the recipient environment, and they should be able to establish initial populations, and those populations should be self-sustaining populations without any additional introductions. There are lots of effects of AIS, such as loss of genetic diversity, parasitic infections, and competitors for food and space, and they also cause problems with infrastructure. Two recent examples are zebra mussel and New Zealand mud snail, which makes severe economic damages such as clogging the water pipes. To manage invasive species worldwide, people are spending billions of dollars, and in North America, most of the cost is to manage aquatic invasive invertebrates. However, the management strategies could hamper due to the existence of cryptic species complexes, meaning morphologically similar but distinct species. We might be treating wrong species, thinking it as a singular species, but in fact they are multiple species, which will lead to loss of time, money, and effort of these management strategies. Therefore, correct identification of the species is the key for successful management strategies. The species that I am interested in is such an invader, which is Viviparous georgianus. It is now known as 𝘝𝘪𝑑𝘪𝘱𝘢𝘅 georgiana. It is actually an interesting aquatic snail because it is native to southern US but invasive to northern US, and it was first introduced as an intentional release to Hudson River. They have olive green shells and have brown bands in their body. Wijesooriya is a problematic species because it is a voracious predator of largemouth bass eggs, and it is a threat for recreational fishing. It also competes with native species for food and space, and is also a host for different varieties of parasites which could be harmful for human and wildlife. In our sampling period, we were mostly able to see invasive snails rather than the native snails, which is not a good sign. According to Cato, these snails is a part of a cryptic species complex with Viviparous limmi and Viviparous 𝘗𝘓𝑒𝑖 in its native range. But we do not know whether it is same to its invasive range in northern US. Therefore, my problem statement will be, does Wijesooriya belong to a cryptic species complex in its introduced range? The viviparous species that I have mentioned here, like Viviparous limmi and Viviparous contactus, looks very similar to Wijesooriya I found in the New York Great Lakes Basin. So we need to confirm whether we are dealing with a single species or multiple species that are just looking similar to each other, causing problems in identification. For example, the picture I took from Wijesooriya et al. 2019 have different viviparous species in it, including Viviparous georgianus. And it seems very difficult to identify the differences between these species. So, my aim is to determine the presence of cryptic species complexes in invasive snail Viviparous georgianus. My samples were collected mostly from New York Great Lakes Basin area, and the major sampling sites were in the Raquette River, which is connected to St. Lawrence River, and more distantly with Lake Ontario. Raquette River is one of the most fragmented rivers in the east of the Mississippi River and is the second longest river of New York State. It is a perfect study site to identify any anthropogenic barriers like dams and vectors like ports, which are important for the dispersal of these species. Now, morphological differences could happen due to environmental variations such as water chemistry, and they could play a major part. And another reason is introduction of multiple species which are looking similar to each other. In other words, existence of cryptic species complexes. Therefore, I hypothesized that different populations of invasive Wijesooriya snails will show differences in shell morphology among the populations we collected, and also Wijesooriya will exhibit distinct phylogenetic subdivisions in its introduced range. For my experimental design, we collected snails by doing field work from May to August in 2021 and transported them to the lab at Clarkson University. Next, we took photographs of each snails and took initial morphological measurements such as shell length, shell width, aperture length, aperture width, and the wet weight of the snails. Next, we conducted a statistical analysis of shell measurements using SPSS. After morphometrics, we extracted DNA using head tissues of each snail, and next we sequenced each snail using the mitochondrial barcode marker, which is cytochrome C oxidase gene fragment. I verified the CO1 sequences I received using GenBank, and next I used software bioedit to align and edit the CO1 sequences. For the phylogenetic analysis, these aligned CO1 sequences will be used and a phylogenetic tree will be constructed using Mega software. I will use sequences available from other studies and sequences for the cryptic species complexes in the native range as reference sequences in the phylogenetic tree. The phylogenetic tree will be then used to check for reciprocal monophyly in the sequences along with the genetic distances to identify existence of cryptic species complexes. The preliminary results I have indicates the box plots generated in SPSS for Wijesooriya, and it visually represents the significant differences between the shell measurements across the populations. In the x-axis, there are different populations from different water bodies. To confirm the morphological data, next we looked at the statistical test done in SPSS. The Manoa test in SPSS gave us p-values lower than 0.05, which are highlighted in yellow here. It confirms that the differences that we are seeing in the shell measurements are actually significantly different from each other. But as a matter of fact, these differences could be either due to environmental variations or due to existence of cryptic species complexes. To confirm that, we will need the phylogenetic analysis results, and hopefully in the future I intend to solve that problem soon. Thank you for listening.